Welcome everyone to our FDA Clinical Investigator Training course, familiarly known as CITC 2022. Good day, good afternoon, or good night, depending on where you may be. It is so great to see so many of you online with us here today. We certainly appreciate your commitment in attending this event from wherever you may be. And taking a peek at the chat pod earlier, I see we have wide global representation. What an utter delight to see such vibrant participation. And, just to name a few, I can't name them all. We have representation from Ghana, Sudan, Norway, Portugal, Poland, India, Canada, Egypt, Switzerland, Philippines, Turkey, Spain, Israel, global, as I said. And right here at home, we have representation from the north, south, east, west, and all in between. This is so fantastic. I hope you guys are excited as I am excited. My name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance, SBIA program, which operates within the Division of Drug Information, DDI, which in turn resides in FDA's Center for Drug Evaluation and Research's CDER Office of Communications. It is my extreme pleasure, along with my FDA colleagues, to be with you here today. This 2 half-day conference features an unsurpassed cast of subject matter experts discussing a variety of topics which cross-cut centers and which was specifically and handpicked and designed for this audience. We entreat you to make the most of this experience, attend all the sessions that your schedule allows, learn all that you can, and submit your questions during the available Q&A sessions. And of course, don't forget to network in the chat pod. In addition to the Adobe Connect platform, we are offering live YouTube streaming. So please invite your colleagues. However, please note that only those attendees who register and attend via Adobe Connect will be able to submit questions to our speakers. And as we set up here for the poll, and let me expand my screen so I can see the results. As we set up here for the poll, which I try to do each conference, I know that CITC has established a very dedicated audience throughout the years. And we would like to get a feel as to how many of you, once having been introduced to SBIA via CITC, how many of you may have attended any other SBIA events? So here's the question. Please make every effort to participate if you can, as we would like to get as many and as accurate numbers as possible. The question is, how many SBIA events have you attended? The choices are A, this is my first one, B, 2 to 5, C, 6 to 10, D, more than 10, and E, most since 2010. Oh, I'm loving these numbers. Wait a couple seconds here to see if some of the numbers stabilize. Okay, let's take these numbers here. For those of you who selected, this is my first one, 63% of you. Oh, that's fantastic. I really hope it does not stay your first one. And this is a fantastic event to attend as your introduction to SBIA. And I hope, as I said, you'll be with us again, and next time you'll be able to select B. Oh my, we have one brave soul who has attended almost all of our SBIA events since they started in 2010. Thank you so very much for being so dedicated. Okay, let's see. As of this morning, we have the very impressive number of over 9,075 registrants representing 122 countries. Thank you all for such an overwhelming response. It's very reassuring in knowing that, we are hitting the target audience. Over 47% of these registrants have self-identified as small businesses. And of those, over 25% report having 1 to 9 employees. These stats continue to be extremely important to us. As I said, they confirm that we are indeed reaching the small business sector of our audience. And they also validate our program's mission and the very essence of the program itself. This is a snapshot of our geo map for today's event, and you can actually click on this image, which will take you to your browser, where you can zoom in and have a closer look at our global participation. So have fun with it. This slide here provides the QR codes for our social media avenues. You can scan them with your phones and be taken to the appropriate platform. And they will also be advertised across the screen banner during our breaks if you miss it now. Be sure to sign up for email updates so that you can be alerted to any upcoming events. And I'll just do this here. So let's turn our attention to some logistics. In our ongoing environment, this event is 100% remote, in addition to being 100% virtual. As such, there may be some internet issues, but please bear with us, and if in fact we encounter those, we will try to get them resolved as quickly as we can. And speaking of tech issues, here is a link for our one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. It's the same page that you used this morning for your online access to the event, so please bookmark that page, as you may need it during the day and tomorrow too. If you have any technical questions about this conference, please check there first, and you will most likely find your answer. We invite you to submit your questions to us so that we can have a vibrant and productive Q&A segment, which is the life force to these events, in my opinion. This is a learning opportunity, and I encourage you to take full advantage of the expertise we offer here. You won't find it anywhere else, I can tell you that. We will answer as many questions as time allows, and when you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name, as shown here. Also, during each presentation, there's a link on the right side of the pod where you will see the speaker's headshot, and it says download the slides. Note that this link is only active during the real-time presentation, and the link actually takes you back to the one-stop shop site so that you can get the slides from there. There will also be a link to a speaker evaluation under the speaker's picture or information on the right side of the screen. We encourage you to take the time to complete the evaluation, as your feedback is much appreciated by the individual speakers. In addition to the speaker evaluation, we also have a final event evaluation or survey. Upon completion of this survey, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The certificate must be downloaded immediately upon completing the survey, otherwise that particular opportunity is lost. Even if you do not want or need a certificate, please take a few minutes to complete the conference evaluation, as your feedback will help us at SBIA and will guide the design of our future events. Note that both the evaluation and certificate are only available for 2 weeks, which means the deadline date is December the 22nd, 2022. The survey link will be posted on our one-stop shop page this afternoon. And as a reminder, all of the sessions are being recorded. It will take us a week or so, business days, to process the recordings. And in the interim, the YouTube link for each day's recording will be posted on the One-Stop Shop webpage at the end of the day. However, you will soon be able to access them from SBIA's homepage, and I hope that you have already bookmarked our page, which is www.fda.gov/CDER SBIA. That's CDER SBIA. That page is in fact CDER's main industry entry page, and it features a treasure trove of regulatory references, our SBIA events calendar, and our SBIA Learn repository with over 748 searchable webcasts. We are also extremely pleased to announce that this conference, in its entirety over the 2 days, offers a total of 7.75 contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process and who attend via Adobe. Please note the emphasis on real-time and Adobe Connect. No CEs can be given for those who attend via the direct YouTube links or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers, and all of the relevant financial relationships listed for these individuals have been mitigated. The planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to the attendance and complete the final activity evaluation via the CE portal, that's ceportal.fda.gov. For multi-day activities, participants must attend to their attendance and complete a faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity, no exceptions. Again, the deadline for this 2-day event is December the 22nd. Please remember that date. I apologize in advance, but we are unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. Your NABP profile number should be the 6 to 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month date format, example 1207. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. Physicians and nurses may then view, print their statement of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of the activity to obtain their CE credit. So if you log in 1 week's time, please note that it may not be there. You may have to wait a full 8 weeks. To simplify things, each night SBIA will send one email to the attendees of that individual event day, and that email will have both information on the CE claim codes and the survey link. Little time left, so let's take a couple minutes to highlight our upcoming events. On December 14th, we'll have a 1-hour webinar, What to Expect After Inspection, 483s, Responses and Beyond. This will focus on compounding inspections. Then on January 13th, we'll have a 1-hour webinar on reporting individual case study reports to FDA Adverse Event Reports System using ICH E2B R3 standards. And I think that particular webinar may be of interest to this audience. Then on January 26th, another 1-hour webinar, FDA's Labeling Resources for Human Prescription Drugs. More information is available on our homepage, seen here, which you need to bookmark. And we also invite you to visit our CDER Learn webpage, where you can link to SBIA tutorials, along with other educational resources, many of which offer healthcare professional CEs. And of course, that website is www.fda.gov/CDERLearn. At this point, I would like to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit. And special thanks to Drs. Leonard Sachs and Millie Dugal for designing the agenda, recruiting and interacting with all our subject matter experts. To CDER for all the supportive roles that they play in bringing this event to fruition. To our dedicated speakers, panelists, and SMEs for their commitment and time. To our DDIers who respond to inquiries each and every day. And most of all, to you, the audience. You the ones who have created the demand for this event. And now I'm sure you're very excited for us to launch fully into today's agenda. I am extremely honored to introduce our host for the next 2 days, Dr. Leonard Sachs. Dr. Sachs is the founder of CITC, one constant and the driving force behind it since it first launched over 10 years ago. He joined FDA in 1998 as a medical reviewer in the Office of New Drugs. Subsequent positions including Acting Director of the Office of Critical Path Programs and Associate Director for Clinical Methodology in the Office of Medical Policy in the Center for Drug Evaluation and Research. In this capacity, he has led efforts to support novel approaches to clinical trials, including the use of electronic technology. Besides his involvement in the design and analysis of clinical trials, he maintains a special interest in tuberculosis and other tropical diseases and has published and presented on these topics. Please help me to welcome Dr. Leonard Sachs.
Thank you very much, Brenda. And like you, I'm delighted to welcome everyone to FDA's 2022 Investigator Training course. As Brenda mentioned, the course this year remains completely virtual, and that has forced us to shorten some of the sessions and to shorten the number of the sessions. But we still hope that we've been able to select the most suitable topics that are both relevant and current. We indeed have a wonderful faculty of FDA lecturers covering the essentials of clinical trials. We'll be covering issues of safety and efficacy, subpopulations, the analysis of results. We have a session on non-clinical studies and the investigator's brochure. We cover compliance, other selected topics of importance, such as new therapeutics, innovative trial approaches, and international trials. All the sessions are followed by live panels, so please send us your questions in the chat and we will try to respond to as many as we can. We very much hope that you will find the content informative and useful, and we are looking forward to seeing you in person in years to come. So for the purposes of context, I'm going to spend the next 15 minutes giving a quick overview of FDA, its structure and mandate, and then we will move on with the first session. In order to contextualize our course, this short talk is on the structure and mandate of FDA. I'm going to cover a brief history of FDA, explain the regulatory processes for approval and clearance of drugs and medical devices, and describe the roles of the Code of Federal Regulations, guidances, and advisory committees. Why do we have regulatory agencies? They're generally built on a legacy of public health failures. Safety, for example, and the illustration in the middle shows sulfanilamide elixir, which was a drug that was marketed in 1937. It contained diethylene glycol as a solvent, a very toxic compound, and resulted in the deaths of 107 people, including many children. And that was the basis for many of the new safety regulations. You can see at the top examples of the phocomelia that resulted from thalidomide use in the 1960s, and many other regulatory agencies introduced safety regulations after that. The drug was never approved in the United States, thanks to the diligent work of Francis Kelsey, a reviewer in the FDA who prevented its approval. Other things that have occurred since then are ethics violations. And the New York Times quote there refers to the Tuskegee study of syphilis, where individuals were not given access to effective therapy and there was no informed consent. They were not aware of what was being done to them, and that's obviously been remedied with our informed consent laws. Fraud has been another issue, and counterfeits and adulteration, among others, are reasons why we have regulatory agencies. A quick history of FDA. In 1902, the first legislation was promulgated regarding the control of medical therapeutics. It was the Biologics Control Act. It was followed by the Pure Food and Drug Act in 1906. In 1912, the Sherman Amendment prohibited false therapeutic claims. In 1930, the agency was named the Food and Drug Administration. In 1938, the Food, Drug and Cosmetic Act was promulgated, where it was required to prove the safety of therapeutic products. In 1951, prescription only was codified. And in 1962, the Kefauver-Harris Amendment required the proving of effectiveness of new medical products. And since then, there have been many user fee acts, as you can see there, PDUFA for prescription drugs, MDUFA for medical devices, JDUFA for generics, and so on. Today, FDA is a huge organization. It has more than 18,000 employees, and it's estimated to regulate 20 cents per each dollar spent by US consumers. And in the picture, you can see our premises at White Oak, Silver Spring. The FDA budget is helpful in giving us an idea of how different activities are resourced. Out of a total of $6.1 billion, you can see that the lion's share goes to human drugs, depicted in orange in the pie chart, 33%. Devices and radiological health, 10.4%. Biologics, 7.2%. And these are the three areas of human therapeutics that this course will focus on. So just to make these a little clearer, drugs, these are small molecules, generally synthetic. They're analytically simple, they're heat stable, and the regulations pertaining to drugs are in 21 CFR 300. Biologics are generally large molecules. They derive from living organisms. They're analytically much more complicated. They involve vaccines, gene therapy, tissue and blood products, other cellular products. They're generally heat labile, and the regulations are in 21 CFR 600. And finally, devices. These are manufactured. The considerations are engineering and physical considerations, and they involve things like catheters, prosthetics, pacemakers, defibrillators, and in vitro diagnostics. And the relevant regulations are in 21 CFR 800. This gives you an idea of what the Code of Federal Regulations looks like. This is the section pertaining to drugs, and you can see Part 312 deals with investigation of new drug applications, or INDs. Part 314 deals with new drug applications, or NDAs. So an investigational new drug application is primarily designed for the protection of human subjects. It's required in the US in order to initiate human studies, and technically the IND allows shipping of investigational drug across state lines for the purpose of conducting a clinical trial. And the purpose of the IND is to ensure that studies are safe and ethical, that they're likely to produce meaningful results, and that there's satisfactory monitoring and reporting of safety. There are situations where a study may be exempt from an IND. These include when the drug is a product that's lawfully marketed in the United States, when it's not intended to support a new indication, a significant labeling change, significant change in the advertising or commercialization of the product, where it does not involve a route of administration, dose, patient or population, or other factor that significantly increases the risk, and where the investigation complies with IRB and informed consent regulations. And the citation is listed at the bottom. FDA may put a clinical study on hold when there's an unreasonable and significant risk of injury or illness, when the clinical investigators are not qualified, when there's insufficient information to assess the risks to subjects, when there's strong evidence of lack of effectiveness, when the investigative brochure is misleading, erroneous, or materially incomplete, when the protocol design is deficient to meet its stated objectives. Studies can also be put on clinical hold when they interfere with other adequate and well-controlled studies, and when drugs with better potential benefit-risk balances are available to the same patients. A new drug application, or biologics licensing application, is the package that's submitted to FDA for review in order to gain marketing approval. There's a list of required components in the CFR, including safety reports. The NDA includes, for example, non-clinical studies, chemistry and in vitro studies, animal studies. It includes efficacy and safety results from clinical studies that are performed under investigational new drug applications. The 1572 is a contract between investigators and sponsors, and in the 1572, these are the conditions that the investigator agrees to. The investigator agrees to comply with the protocol, to personally conduct or supervise the investigation, to obtain informed consent and IRB review, to report adverse experiences, to read and understand the investigator's brochure, to ensure study staff are aware of their responsibilities, to maintain adequate records and make them available for inspection, and finally to ensure IRB oversight and notify IRBs of problems or changes. I mentioned the investigator's brochure, and this is a document that includes description of a drug substance and formulation. It has a summary of pharmacology and toxicology in animals and, if known, in humans. It has a summary of prior information of safety and effectiveness in humans. It addresses the possible risks and the side effects of the investigational product, and it also includes precautions and special monitoring that may be needed. And on the basis of the investigator's brochure, you would be making a decision if the study is safe and appropriate for your patients. This slide shows the different types of NDA submissions. A 505(b)(1) involves a full development program with all phase 1, 2, and 3 data. A 505(b)(2) is a submission where at least some of the information comes from studies not conducted by or for the applicant. For example, they may come from the literature or by reference to relevant previous FDA findings. A 505J is the generic pathway where the product is shown to be identical to a previously approved product based on chemistry and bioequivalence. Subpart H relies on a surrogate endpoint that is reasonably likely to predict clinical benefit or an effect on a clinical endpoint other than survival or irreversible mortality. It's been used in the beginning for HIV drugs and subsequently for many other applications. And finally, the animal rule is used when efficacy studies are not ethical or feasible, for example, in the prophylaxis of anthrax. CITC has a number of programs to expedite drug development for serious conditions in need of new treatments. Accelerated approval is based on the effect on a surrogate endpoint or an intermediate clinical endpoint that's reasonably likely to predict a drug's clinical benefit. Priority review is given to drugs that satisfy a serious unmet medical need, and in those situations the review is completed in 6 months instead of the 10-month standard review. Fast track, again, is a method for FDA to take actions to expedite development and review. And finally, breakthrough is a designation that's given to products that, based on preliminary clinical evidence, are promising in providing therapy for serious diseases where available therapy is not adequate. And as part of that program, FDA gives intensive guidance on efficient drug development, makes an organizational commitment, allows a rolling review, and takes measures to try and get the drug to patients as soon as possible. A couple of words about devices. An investigational device exemption is equivalent to the IND in the drug world. It ensures protection of human subjects in clinical trials. It's needed for significant risk device studies, for example, for in vitro diagnostics, when the result significantly affects patient treatment in a way that presents a serious risk. And even if an IDE is not needed, informed consent and IRB review are often necessary. Classification of medical devices, unlike drugs and biologics, they're divided into 3 classes based on the intended use and associated risks. Class 1 are low risk devices, and they require general controls. These are things like bandages and manual stethoscopes, and they're exempt from pre-market submissions. Class 2 are moderate risk devices. These require general and special controls, and they include most laboratory tests, local knee replacements, total knee replacements, powered wheelchairs, and so on. And a 510K is the most common pathway to market. And Class 3 devices are the highest risk devices. They require general controls and pre-market approval. And they include things like implantable pacemakers, spinal disc replacements, and they require a full pre-market approval package. As far as pathways to approval or clearance of devices go, the 510K relies on substantial equivalence to a predicate device. The de novo pathway is used where a predicate device does not exist, and a pre-marketing approval or PMA is needed for Class 3 devices and new devices where the risk cannot be mitigated by special controls. And valid scientific evidence must be presented to demonstrate a reasonable assurance of safety and effectiveness. A word or two about harmonization. Obviously, in the world of global trials and drug development, harmonization with other regulatory authorities is really critical. The International Council for Harmonization was established in 1990, more than 30 years ago. The goal is to produce consensus documents that support global approaches to drug development. There are approximately 40 guidelines listed on their website, and E6 is the guideline on good clinical practice, which deals with the quality of clinical trials. The International Coalition of Medicines Regulatory Authorities is an informal coalition of regulators which focuses on aligning regulatory recommendations, has more than 30 participating countries. And then besides these harmonization efforts, we have interagency agreements between FDA and other agencies allowing discussion and consultation. And our guidances and regulations are all released in draft. They're announced in Federal Register notices, and dockets are open for public comment. So this is an attempt to make sure that there's general public input into the activities of the agency. How does FDA make its decisions? Primarily based on scientific review, also on the requirements of the Code of Federal Regulations, on guidances, and on the advice obtained from advisory committees. Review teams may involve several different disciplines, including chemistry, clinical pharmacology, toxicology, microbiology, clinical reviewers, and statistical review. These are examples of guidance documents, numerous guidances which address many different aspects of drug development, and we certainly recommend referring to these when engaging on a drug development program. This is an advisory committee. Advisory committees make use of a panel of external subject matter experts, and they're an important tool for FDA to ensure that the broader scientific community's opinions are incorporated into our decisions. Finally, product labeling. This is probably one of our more tangible products. The product label contains information including approved indications and use, dosage and administration, warnings and adverse reactions, drug reactions, use in specific populations, and it contains a description of important clinical studies. It's used by healthcare professionals and patients for information on safe and effective use, and it has important implications for advertising and for promotion. To end off, a couple of questions. First, an IND can be waived for a drug not approved in the USA, but approved in other countries. True or false? That's false. INDs can only be waived for drugs that are approved in the USA. Under what circumstances can the animal rule be used? Well, as you'll remember, the animal rule is there for situations where a drug cannot be tested safely or ethically in humans. What is accelerated approval? Accelerated approval is based on a surrogate endpoint or an intermediate clinical endpoint. And finally, drug labels contain data on clinical trials that supported approval. True or false? And that's true. Section 14 of the drug label typically reports a couple of studies that were important in the approval process. So at this point, we're going to move on with our program, and I'm delighted to welcome my dear colleague, Karen Hicks, who has many years of experience in FDA and many years of experience as an interventional cardiologist. And she's going to tackle the first important issue in clinical trials, which are endpoints focusing on cardiovascular trials. So I'd like to hand over to Karen.
Thank you for that introduction. It's great to be here. Over the next 30 minutes, I'll be discussing the most important things you need to know about endpoints in cardiovascular trials. I have nothing to disclose, and the opinions expressed here are my own. Here are the learning objectives for this presentation. I'll discuss efficacy endpoints, including important information you need to know about primary and secondary endpoints, describe frequently used endpoints in cardiovascular trials, define and discuss surrogate endpoints and biomarkers, and describe the basis for traditional approval and accelerated approval. Before I get started, I'd like to share the following two publications with you. In 2009, FDA established the Standardized Data Collection for Cardiovascular Trials Initiative. The goal of this initiative was to simplify the design and conduct of clinical trials intended to support marketing applications. A large global effort, this initiative included over 20 stakeholders, including representatives from some of the best clinical events committees in the world. Together, we created cardiovascular and stroke endpoint definitions for clinical trials. These definitions have been used not only in diabetes and cardiovascular trials, but also in many other therapeutic areas. These definitions and data standards have transformed how clinical trials are conducted and data are collected at the FDA. When you download this publication, please be sure to also download the appendix, which includes the definitions. Here are the endpoints we defined. If you go to the appendix, you'll note that cardiovascular death is classified into 7 categories, and non-cardiovascular death is classified into 16 categories. This publication provides the data standards for these definitions. So now let us turn our attention to efficacy endpoints. An efficacy endpoint is a measure designed to reflect the intended effects of a drug. Efficacy endpoints can include assessments of clinical outcomes such as death, myocardial infarction, and stroke, symptoms such as pain, dyspnea, and symptoms of depression, functional measures such as the ability to walk or exercise, and surrogate endpoints, which I will discuss more later. The primary endpoint is the endpoint or endpoints that establish the effect of the drug and will be the basis for concluding that the study meets its objective. When an effect on the primary endpoint is shown, the secondary endpoints can be formally tested. A secondary endpoint can be a clinical effect related to the primary endpoint that extends the understanding of that effect, or can provide evidence of a clinical benefit distinct from the effect shown by the primary endpoint. In general, it may be desirable to limit the number of secondary endpoints because if multiplicity adjustments are used, the chance of demonstrating an effect on any secondary endpoint may become increasingly small as the number of secondary endpoints increases. Or if a hierarchy is used, the important hypotheses further down the hierarchy might never get tested. Primary and secondary endpoints need to be included in a plan to control the overall type 1 error. Exploratory endpoints do not need multiplicity adjustment because they are generally not used to support conclusions. Efficacy endpoints can be single or multiple. When you have multiple endpoints, multiplicity issues can arise. This slide summarizes types of multiple endpoints. With co-primary endpoints, you need to demonstrate an effect on each co-primary endpoint. With several primary endpoints, you need to demonstrate an effect on at least one of the primary endpoints. Major adverse cardiovascular events, otherwise known as MACE, is one of the most frequently used composite endpoints in cardiovascular trials. You want to use this composite endpoint in a trial when more than one clinical outcome is important and all outcomes are expected to be affected in some way by the treatment. It's also defined as the first occurrence of any one of the specified components. So it's a time to first event analysis. So if you're using MACE, it's the time to the first event, whether or not it's cardiovascular death, myocardial infarction, or stroke. Sometimes a critical endpoint occurs too infrequently to be used as a primary endpoint. For example, death. So it is important to include the event in a composite endpoint for the primary endpoint and as a planned secondary endpoint, especially if you are seeking a mortality claim. Here are results from the Plato trial in patients with acute coronary syndrome. The composite endpoint was MACE, cardiovascular death, non-fatal MI, and non-fatal stroke. Ticagrelor decreased the risk of the composite endpoint by 16%, which was statistically significant. The difference in treatments was driven by cardiovascular death and myocardial infarction. Note that cardiovascular death and all-cause mortality were also included as secondary endpoints and were statistically significant. So this is an example where cardiovascular death was included in the primary endpoint, but also as a secondary endpoint, and all-cause mortality was included as a secondary endpoint. This is a time to first event curve, also known as a Kaplan-Meier curve for the composite endpoint. So days from randomization are listed on the x-axis, and on the y-axis is the cumulative percent of the composite endpoint that has occurred. So what makes a good endpoint? First, you want to select an endpoint or endpoints upon which the drug is thought to have an effect. So if you have a P2Y12 inhibitor, based on the mechanism of action of the drug, you would expect this drug to affect thrombotic events such as CV death, non-fatal MI, non-fatal stroke, and stent thrombosis. Second, you want to consider the population. If you study ACS patients, you're going to get a lot of events, especially within the first 30 days of their myocardial infarction. So if you have an event-driven trial, you're likely to get the number of events that are needed because the population is enriched. ACS trials are generally going to be at least a year or so in duration. Atrial fibrillation trials may require a longer duration to achieve the number of events needed to show a treatment difference. And for example, in the Aristotle trial, apixaban had a follow-up of 36 months. And in the Rocket AF trial with rivaroxaban, the median follow-up was a little less than 600 days. Heart failure trials with sacubitril valsartan had median follow-up of approximately 2 to 3 years, with total treatment up to greater than 4 years. Finally, timing of the endpoint is critical. You want to make sure the drug effect is optimally captured. If you try to capture the effect too early, you may not allow time for the drug to work, but too late gets confounded by intercurrent events. There are several ways to analyze events. One way is with the time to event analysis, as we discussed previously. This is used for MACE and cardiovascular trials, and it's also used for progression-free survival and overall survival in oncology trials. In some oncology trials, progression-free survival is selected as the primary endpoint, and overall survival is selected as the secondary endpoint, because an effect of treatment on disease progression is clinically important, may be more readily demonstrable, may be detected earlier, and may often be larger because the observed effect on overall survival can be impacted by subsequent treatment post-progression. Change from baseline is used to analyze blood pressure results, depression scores, hemoglobin A1C, and 6-minute walk distance. And finally, clinically meaningful change is important in patient-focused drug development, or clinical outcome assessment, such as patient-reported outcome measures. What are the attributes of a quality clinical trial? Excellent follow-up. From a practical standpoint, you want to avoid loss to follow-up and missing data, which can make a trial uninterpretable despite our best attempts at sensitivity analyses. It's also important to recognize that even if a subject wants to withdraw consent and wants to stop the treatment, there are follow-up options for the subject to stay in the trial. They could stop the drug but continue with follow-up visits, or they could continue to allow telephone contacts and medical records checks. Let's look at some examples of endpoints in cardiovascular trials. For the ACS population, the primary endpoint is typically MACE. For a heart failure trial, the primary endpoint is typically cardiovascular death and hospitalization for heart failure. In some trials, urgent heart failure visits have also been part of the primary endpoint, such as with the DAPA heart failure trial. In non-valvular atrial fibrillation trials, the primary endpoint is typically stroke and systemic embolism. And for pulmonary hypertension trials, these trials can have a single endpoint, such as exercise capacity per the 6-minute walk test, or they could have some type of composite endpoint for clinical worsening, which includes all of these other endpoints. Let's change gears now and discuss surrogate endpoint. A surrogate endpoint is a laboratory measurement or physical sign that is used in therapeutic trials as a substitute for a clinically meaningful endpoint that is a direct measure of how a patient feels, functions, or survives, and is expected to predict the effect of the therapy. Some examples of validated surrogate endpoints include blood pressure, hemoglobin A1C, and LDL cholesterol. Accelerated approval is approval based on an effect on a surrogate endpoint or an intermediate clinical endpoint that is reasonably likely to predict a drug's clinical benefit. However, certain conditions have to be met. First, the illness has to be serious or life-threatening. The drug has to provide a meaningful advantage over available therapies, and post-approval studies are required to verify and describe the drug's clinical benefit. And if these trials aren't conducted, or if the trials are conducted but don't verify and describe the drug's clinical benefit, FDA may withdraw approval of a drug or indication approved under the accelerated approval pathway. There are pitfalls to using surrogate endpoints. The surrogate may not be valid, or the drug may have unexpected, unfavorable effects. There have been some epic fails with surrogate endpoints. If you think back to the 1990s with the CAST-1 and 2 trials, with anconide, flecainide, and marizicine, and how patients post-MI with premature ventricular contractions were being treated with these drug products that actually increased mortality. Another example is torcetrapib to increase HDL, which was also associated with increased mortality. A third example is erythropoietin, which was also associated with increased mortality. So sometimes we think that a surrogate endpoint will predict a clinical benefit, but it actually causes harm. In addition, many times it's unclear what magnitude of change is clinically meaningful. So balancing risk and benefit can be challenging. Reliance on a surrogate is usually an alternative to a large outcome trial, which is not performed until phase 4. What is required to validate a surrogate endpoint? Well, first you need to understand the pathophysiology. You also need to identify a marker upon which several drugs can work by different mechanisms. You need to validate the marker and associate this marker with the clinical outcome. So if we use blood pressure as an example, we understand the pathophysiology of blood pressure. It's used as a clinical endpoint. Epi data shows us that elevated blood pressure increases the risk of stroke, MI, heart failure, and renal failure. And by decreasing blood pressure, we decrease the risk of adverse outcomes. Blood pressure is also assessed by physical examination. Let's talk for a minute about biomarkers. Biomarkers are different than surrogate endpoints because they are not an assessment of how a patient feels, functions, or survives. A biomarker is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. And I also wanted to provide you with this example of how NT-proBNP, a biomarker, was used to gain a pediatric indication with sacubitril and valsartan for the treatment of symptomatic heart failure with systemic left ventricular systolic dysfunction in patients aged 1 year and older. Reductions in NT-proBNP were expected to improve cardiovascular outcomes. And so based on these biomarker results, this drug product was granted a pediatric indication. Let's talk for a moment about the basis for traditional approval and accelerated approval. Traditional approval is based on clinical endpoints that reflect how patients feel, function, or survive, or validated surrogate endpoints, those that have been shown to predict a specific clinical benefit. Accelerated approval is based on a demonstrated effect on a surrogate endpoint that has not yet been validated, but is reasonably likely to predict a clinical benefit, or an intermediate clinical endpoint that is also reasonably likely to predict a clinical benefit. In summary, we've discussed that an efficacy endpoint is a measure designed to reflect the intended effects of a drug. Efficacy endpoints include assessments of clinical events, symptoms, measures of function, and surrogate endpoints. A surrogate endpoint is a laboratory measurement or physical sign that is used in therapeutic trials as a substitute for a clinically meaningful endpoint that is a direct measure of how a patient feels, functions, or survives, and is expected to predict the effect of the therapy. Accelerated approval is based on an effect on a surrogate endpoint or an intermediate clinical endpoint that is reasonably likely to predict a drug's clinical benefit. And finally, a biomarker is not an assessment of how a patient feels, functions, or survives. A biomarker is a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. So based on what you've learned today regarding efficacy endpoints, surrogate endpoints, and biomarkers, I hope each and every one of you will go on to plan a highly successful trial that brings a completely new therapy to market. The next two slides provide you with a list of resources. Okay, it's time for our first challenge question. Take a second to consider which answer you think is correct. Validated surrogate endpoints include all of the following except A, LDL cholesterol, B, blood pressure, C, vulnerable plaque, or D, hemoglobin A1C. Excellent. I think you're ready for the second challenge question. Which of the following statements is false? A surrogate endpoint is A, a laboratory measurement or physical sign, B, used in therapeutic trials as a substitute for a clinically meaningful endpoint, C, a direct measure of how a patient feels, functions, or survives, or D, not expected to predict the effect of the therapy. Take a second to consider which answer you think is correct. Excellent. Thank you for your attention. That concludes my presentation. I will be back shortly to answer questions.
Hello, everyone. I'm really excited to be here today as part of the first session in this year's course. And my topic today is specific populations in clinical trials. This is my disclosure slide. In addition, I want to just point out a couple of things. The first is that my talk will focus on specific populations as defined by FDA. And specific populations is a newer term. You may have heard the older term special populations, but we use specific populations now because we think it's a more appropriate and probably more precise term. The second point I want to make today is that it's going to be a whirlwind talk, and I'm really just going to provide an overview and important guidances that FDA has published recently to provide additional, more in-depth information for you to consider. And then thirdly, that I am a pediatric nephrologist, so while my clinical expertise is limited to pediatrics and pediatric nephrology, I'm going to be talking about several specific populations today. So what do I hope to go over with you in the next 30 minutes or so? I want to review with you the inclusion of specific populations in drug development. And the three populations I really want to focus on today are children, pregnant and lactating individuals, and geriatric populations. In addition, I do want to also discuss enhancing overall diversity in clinical trials. So why am I giving you a talk on specific populations? Let me just say that on this slide, I think it demonstrates the importance that FDA has on collection of information on specific populations. Under the physician labeling rule, which is a regulation that describes, importantly, the content and format of prescription drug and biologic product labeling, there are specific sections under Section 8 that describe important information that should be included in specific populations, and you can see sections 8.1 through 8.5 listed here. I'm going to go over, each of these populations during my talk today, and let me just also note that there are other specific populations that I will not be discussing, and those include specific populations that have hepatic and renal impairment. I'm going to focus today mostly on pregnancy, lactation, pediatrics, and geriatrics. So the first topic I want to review this morning is related to pediatric patients and the inclusion of pediatric patients in clinical trials. So this slide really goes over two basic tenets that are foundational to pediatric therapeutics development. First, that pediatric patients, just like any population, including adults, should have access to medicines that have been appropriately evaluated. And what flows from that is that if a product development program includes adults, but children are likely to use the drug once it's approved, that development program should also include pediatric studies. In the next few slides, I'm going to go over some basic ideas and basic principles that underlie therapeutics development in children. The first of those are the ethical and feasibility considerations. There was a guidance that was just published a couple months ago in September of 2022 on the ethical considerations of clinical investigations of medicinal products or medical products involving children. among other really important concepts brought forth in this guideline are the three basic tenets and three basic principles that you see here. First, that children should only be enrolled in clinical trials if the scientific objectives cannot be met through enrollment of subjects who actually can provide informed consent personally, such as adults. And because children are a vulnerable population, because they cannot provide informed consent, then clinical trials involving children must offer the prospect of direct therapeutic benefit. And without that prospect of direct therapeutic benefit, the risk to the child for enrollment in a trial should be low, and low is basically defined as no more than a minor increase over minimal risk. And those notions and concepts of minimal risk and minor increase over minimal risk are really well defined and described in this guidance. And then finally, the children really should not be placed at a disadvantage by being enrolled in a clinical trial. But even when these when these ethical considerations have been incorporated, there are also feasibility issues and feasibility considerations that are commonly seen in pediatric therapeutics development that are not necessarily as commonly seen in adult product development. And this is, again, a good thing for children because the prevalence or incidence of many diseases and conditions are lower when compared to adult populations. In this next slide here, I want to go over some important concepts that are brought forward in this ICH E11R1 addendum document that I had mentioned a couple of slides ago related to the foundational principles. And in this important addendum guideline that was published by FDA in 2018, there are several areas related to pediatric therapeutics development that are found in this guidance, and it's an excellent guidance to those of you who are interested in learning more about how to conduct pediatric drug development programs. On these three bullets you see next to the guideline, I think I want to highlight these three areas. The first is the use of a strategy called pediatric extrapolation. This can be used to increase the efficiency and feasibility of pediatric product development, and the basic idea behind pediatric extrapolation is that when there are similarities between a pediatric population and a reference population, usually an adult population, that there are similarities in the course of a disease and the response to whatever drug you're studying, that those similarities can be translated into leveraging information that could lead to clinical development that requires less than the standard adequate and well-controlled trial. And this guidance does give some high-level descriptions of pediatric extrapolation. In addition, there is really clear guidance about the enrollment of children stratified or staggered by age. Typically we see, and historically we've seen adolescents studied first, then school-age children, then preschool children, then toddlers, then infants, and then newborns. And the guidance puts forth the important consideration that strategies that are relying on staggered enrollment based solely on age really should be justified, because if there's really no reason to study adolescents only first, there's no safety or efficacy reason to not enroll younger populations at the same time, that one should consider that really because it decreases the time it takes to have the drug studied and therefore then potentially available for children to use. And then finally, the guidance goes over many other unique product development considerations, including age-appropriate formulations and safety information, including the need often for long-term safety information if the long-term safety is unknown in a pediatric population. I want to highlight on this slide one recent guidance that was published again in 2019 about actually including adolescent patients in adult trials. And here the Oncology Center of Excellence published this guidance about inclusion of adolescent patients in adult oncology trials. And really just to suffice to say that there are certain situations in which adolescent oncology patients are very similar, in fact, to adult oncology patients, including young adult oncology patients. And in those situations, adolescent enrollment should be considered to really, again, speed the availability and not delay access of potentially effective therapies to the adolescent oncology patient. And you can see in the second bullet some considerations when it would be appropriate to consider inclusion of adolescents in adult oncology trials. So taken together, I'm just giving you some highlights about considerations related to inclusion of pediatric patients in clinical trials, and just some recent guidance that FDA has published. So moving on, I want to spend a few minutes to talk about the inclusion of pregnant individuals in clinical trials. And as a background, again, no different than children, pregnant individuals should have access to medicines that have been appropriately evaluated. Now, one important distinction between pregnant individuals and children are that medicines for specific indications that are approved in adults are also approved in pregnant adults unless specifically contraindicated. In other words, a pregnant adult is still an adult, and so when the drug has been tested and approved in adults, then one does not need to say that a clinical trial needs to be conducted in pregnant adults to ensure that the efficacy is similar, unlike the situation in children. However, pregnant individuals are often automatically excluded from clinical trials. And we also know that alterations in physiology during pregnancy can affect both dosing and safety in the pregnant individual, that because pregnant individuals are often automatically excluded from clinical trials, there's often little to no human data to support the safety and dosing of the product prior to approval. And that leads to prescribers and pregnant patients really left to make benefit risk decisions without any data. And that situation is really unacceptable. And so recently, FDA has been working with external stakeholders and drug developers to consider what types of information are important to obtain to support, the appropriate dosing and safety of drugs when they're used during pregnancy. One important initiative that was undertaken by FDA over the last 7 years is the Pregnancy and Lactation Labeling Rule, or PLLR. And the reason I bring this up is not to really go into, the changes that occurred in pregnancy and lactation labeling under PLLR. And you can see in this slide, there was a shift in some of the location of the information. But more importantly, the PLLR was intended to improve the communication of risk information that was collected as part of the labeling of a product. But in the implementation of PLLR, FDA learned quite a bit about the state of data availability in drugs used during pregnancy. And in many cases, even though the framework and the new labeling framework was successful in improving the communication of risk when there were data that supported a specific risk, oftentimes what we learned was that data were actually absent, and that even though there was the ability to communicate in a better way, there were no data to communicate. And even when there was data available, often it lacked both quality and quantity. Finally, although there was a great desire to include definitive risk statements in labeling, often there weren't sufficient data, and that's the current state, that there are oftentimes insufficient data to really be definitive about the risks of the drugs when used during pregnancy. So as a consequence to what we learned from the implementation of PLLR, FDA published a guidance on the inclusion of pregnant patients in clinical trials. And this guidance, this draft guidance published in 2018, includes situations when pregnant individuals, pregnant women can be included in clinical trials. It describes and follows the HHS framework for human subject protections. And it really does consider, the inclusion of pregnant individuals both in the post-market and pre-market setting. And finally, an important consideration of this guidance also is what to do when a patient becomes pregnant during a clinical trial. So all of these important considerations are described in this guidance, and I do think that it provides really a roadmap and perhaps a call to consider when pregnant individuals can be safely and ethically enrolled in clinical trials. And let me just say here, too, that we're not proposing that pregnant individuals just automatically be included in clinical trials, because there needs to be important safety information collected before including pregnant individuals. However, I think what this guidance put forward is the idea that automatic exclusion of pregnant individuals in clinical research is also unacceptable. And there has to be a careful review and justification for why pregnant individuals would be excluded. In addition, I think this guidance also sets up important considerations about early discussions and early planning so that the appropriate reproductive toxicity studies can be completed in non-clinical studies so that when the phase 3 trial is ready to begin enrollment, there is already some information that can help guide the safety and the safety monitoring of a drug to allow for pregnant patients to be enrolled in clinical trials. So I just want to spend a few minutes about the collection of important safety information on when drugs are used during lactation. And here again, the problem statement is similar to the problem statements I've already provided you on research in children and pregnant individuals, that medications are used in lactating individuals, often with little to no human data about the safety of the drug, in the breastfed infant. And importantly, collection of data on the transfer of drug into human milk and into the breastfed infant can provide really important information that can help prescribers and patients. Unfortunately, such data are really limited in terms of availability at this time, and generally, almost never do we have the information before the drug is approved. So in 2019, FDA published a guidance on clinical lactation studies. And just briefly here, I want to highlight some of the important considerations in this guidance, including describing scenarios when a lactation study would be appropriate. And also, if you're planning to conduct a lactation study, discusses ethical considerations about who can participate in lactation studies, including lactating women who are already taking the drug for medical or prescription or therapeutic purposes, and also potentially the inclusion of healthy lactating women in lactation studies as well. And those ethical considerations are described in this guidance. There are really, I think, well-described sections about different study designs that can be used and the information that can be obtained in such study designs. And then finally, sampling strategies and pharmacokinetic analysis methods are described. So this is a really, I think, complete guidance for those interested in getting more information on how to and when to conduct clinical lactation studies. So now I want to talk about the last specific population, the inclusion of geriatric patients in clinical trials. FDA has defined as part of their guidance on geriatric information in prescription product labeling as patients 65 years of age and older. And again, here, like pregnant individuals, medicines approved for indication adults are also approved in geriatric populations unless specifically contraindicated. Now, it's important to consider the inclusion of geriatric patients for many reasons, and certainly the three that I've highlighted here on this slide are major considerations. That comorbidities that occur more commonly in geriatric patients could have effects on the efficacy and safety of the product. For example, organ dysfunction can play a greater role in geriatric patients because more geriatric patients, for example, may have organ dysfunction. There's certainly a higher incidence of concomitant therapy and therefore a higher risk for drug interactions. And simply that the geriatric population is increasing in the United States, and therefore, in order to appropriately study drugs and patients are going to use them, if there are more geriatric patients in the United States, then there are likely going to be more geriatric patients taking any given medication. So, importantly, the labeling guidance describes when geriatric information should be placed in that section 8.5. And the labeling should provide information, if any are collected, on differences in the safety, effectiveness, or dosing, that is pharmacodynamics and or pharmacokinetics, in a geriatric population compared to a non-geriatric adult population. Here again, the Oncology Center of Excellence leads the way and has recently published a guidance in March of this year on the inclusion of older adults in cancer clinical trials. Again, outlining why it's important to study geriatric patients in oncology trials, and you can see the at least three reasons here. The guidance also goes over ways to incorporate geriatric patients in trial designs, and also provides approaches to improve recruitment and retention of geriatric patients, and also gives additional information on important and specific adverse event monitoring and management in geriatric patients, and the potential need for collection of additional post-marketing data when geriatric patients are enrolled in clinical trials. But here, really all intended to improve the generalizability of cancer treatments to include all the populations who might benefit from its use, including the geriatric population. I want to spend the last few minutes talking about another important topic that's not specifically related to specific patient populations, but really overall diversity and enhancing diversity in clinical trials. Our current FDA Commissioner, Dr. Rob Califf, has made this really important quote when he said, "The U.S. population has become increasingly diverse, and ensuring meaningful representation of racial and ethnic minorities in clinical trials for regulated medical products is fundamental to public health." So FDA is working to ensure the public health of the whole public, and that really in his quote there, I think, is implied that trials should reflect the population that is most likely going to use the drug. And despite this, certain groups continue to be underrepresented in many clinical trials. So FDA has put out two recent guidances that describe enhancing diversity in clinical trial populations. Their first guidance, published in 2020, includes strategies to broaden eligibility criteria and also provides guidance on inclusive trial practices. And you can see here many topics discussed in the guideline, the guidance that go over ways to enhance diversity, from clinical trial design and methodologic approaches to enrichment strategies, And then importantly, decreasing burden and enhancing retention, both are important, not only to get patients enrolled, but also to keep them in the trial once they are enrolled. And there is finally a section on rare disease considerations. So again, a really important guidance that describes how clinical trials can be potentially more diverse and inclusive of populations who are likely going to be taking the drug. This slide goes over, I think, an important development in improving diversity as part of drug development, drug and device development, and it is a joint guidance sponsored by no less than 5 centers within FDA, and it discusses the inclusion of formal diversity plans as part of drug development. And you can see here that in the opening of an IND or IDE, that a diversity plan is recommended when studies that are being conducted under the IND or IDE are intended to support a marketing application or device marketing application. Diversity plans should be submitted early so that they can be discussed with the agency and advice given no later generally than end of phase 2. And also provides detailed description of what should be included in the diversity plan. I'm not going to go over each of the items here. It's really beyond the time that I have, but I have listed them here so that you can get a flavor for what types of information should be included in a diversity plan. I really think that the inclusion and the recommendation to include diversity plans in all IND or IDs that are intended to move to a marketing application will really move forward the inclusion of appropriate and diverse populations in drug development. So finally, I want to briefly mention the role of decentralized clinical trials in enhancing diversity in product development. A decentralized clinical trial is really just a clinical trial where some or all of the trial-related activities can occur at locations separate from the central or principal investigator's location. You can see here several potential benefits that can enhance diversity, including avoiding having to travel long distances or take off work or expend additional costs to participate in a clinical trial. Those certainly you can imagine, would enhance diversity, particularly in populations where it is difficult to participate because of socioeconomic reasons. In addition, it can also help for conditions that are rare or where the diseases are sporadic, so that those assessments can be made very close to the time and the place where some of the symptoms are occurring. Now, of course, importantly, there are challenges related to decentralized clinical trials, including the need for more staff, because you're just going to have collection of data at more and disparate sites, and that can lead to compliance issues as well. So there needs to be really good documentation, really good training to make sure that every site in a decentralized clinical trial is collecting information in the same way, and that the data that can be used are valid and reliable. Nevertheless, again, I can't overemphasize, I think, the potential utility of a decentralized clinical trial in enhancing diversity. So I hope to have provided you a high-level summary of the inclusion of specific populations in clinical trials for drug development. And on this summary slide, I have 4 takeaways that I think are important for you to remember. The first is that pregnant and lactating and children and geriatric populations all should have access to medicines with information to support their safe and effective use. Secondly, that drug development should be designed to include specific populations when use is expected in these populations. And certainly there are unique ethical considerations in both clinical trials for children as well as pregnant individuals. Early planning is the key to making sure these specific populations are included appropriately. And remember that the U.S. population is really becoming increasingly diverse, And it's really important that we all consider how to be inclusive in clinical trials so that the clinical trials and the data that are obtained from those clinical trials can be generalized to reflect the population that is most likely to use the drug. And finally, the challenge question. Specific populations that should be considered for enrollment in clinical trials during drug development include A, pediatric populations, B, pregnant and lactating populations, C, geriatric populations, D, all of the above, or E, A, and B only. And hopefully you've all gotten the correct answer, which is D, all of the above. I want to thank you for your attention, and I look forward to the question and answer period at the end of this session.
Well, thank you very much, Karen and Lynne. First of all, just a quick apology. I missed my cue and didn't have a chance to introduce you, Lynne. So in retrospect, I just wanted to thank you for being a wonderful representative on the topic of specific populations. And we have a lot of questions now in the chat that have been submitted. And I'm just going to do my best to field them as they come in for individual speakers. So let's see. There's a question for Karen about the Plato trial. The person is asking, what was the benefit of including cardiovascular death as a primary and secondary endpoint in the Plato trial? Any thoughts on that, Karen? I'm just wondering whether we have Karen on. It looks like she's joining us now.
Yes, hi, and welcome. What a turn for this meeting today. Yes, so the reason I included that example is because when you have a composite endpoint, like CV death, non-fatal MI, and non-fatal stroke, if your trial is successful and is statistically significant for that primary composite endpoint, that is the indication you will get in your label, okay? Reduce the a composite endpoint of CV fatal MI, non-fatal stroke. But if you're interested in going after a mortality claim, and again, death might be one of those infrequent events. And if you're using a composite endpoint, you want to include death in the primary endpoint, but you also want to include it as one of the secondary endpoints. So it's included in an overall plan control type 1 error. And that we claim, and Plato did at that time. Thank you.
Great, thanks, Karen. There's an excellent question for Lynne, which really gets to the heart of specific populations. The question is, does specific populations mean vulnerable populations in clinical trial? Lynne, any thoughts on that? Lynne, were you able to get that question? I guess we're having a bit of trouble hearing Lynne at the moment. Or perhaps
Leonard, I think she might be having connection difficult moment. Should we go to a couple of other questions? Hopefully she'll be able to join.
Absolutely, absolutely, Karen. I'm just trying to look at another one. Perhaps I'll take one which was directed at me. the question was how does the FDA regulate repurposing of drugs which are already approved and on the market? I think that's a really good question, because repurposing is obviously a very important strategy to satisfy areas of need where new drug development hasn't occurred. So there's really no particular regulatory pathway. Our regulatory standard for repurposed drugs is just the same as for any others. In other words, they have to provide substantial evidence of efficacy and safety based on well-controlled studies and so on, according to our regs. On the other hand, repurposed drugs have a considerable advantage in that there's already a repository of data and experience. So it's through a 505(b)(2) application, which I mentioned in my introduction, it may be possible to reference all the preclinical work that was done to support the approval of those drugs. It may be possible to reference previous safety data. So the package would be considerably smaller. And of course, if the efficacy data for a new use of an existing drug was available from other sources, for example, literature, that could also be referenced in a 505(b)(2). Let's see if we can get on to another one. I'm just getting a sense, I think Lynne's still having her connection problems. So let's have another one for Karen. There's a question about time to event analyses for medical devices. I'm not quite sure if that's something that you wanted to address, Karen, maybe just from your experience.
Oh, yeah. Yes,
Yeah.
I saw there were two questions, and so time to event analyses can be done not only for drugs, but also for devices. You just have to think about, what is the primary endpoint? So if the primary endpoint is, 12-month target lesion failure, which is a composite endpoint, you're basically doing time to event for the very first event that would, and so just like you can do Kaplan-Meier curves for drug trials, you can as well. And thank you very much for that question.
There's another question, breakthrough. The question is, please clarify how the burden of conducting a US IDE trial for a therapeutic medical device that has US FDA breakthrough designation and is approved outside the US with a CE mark and other country-specific regulatory approvals. I didn't quite get that, but I think the question is how breakthrough applies to devices. So I'm not an expert on device regulations, but the breakthrough program in CDER is limited to drugs. And I think we may have an opportunity later on in the program to ask somebody with more experience with device regulations, but breakthrough is really a drug-specific initiative. Again, just checking, unfortunately still having trouble with Lynne's connection.
Hey, Leonard, I think I'm on.
Oh, excellent, yeah.
Sorry, I'm really sorry to the whole audience about that. Of course, it worked all morning until we had to do Q&A. My apologies.
That is predicted. If you can get your camera on, even better. Lynne, I'm going to try and find a couple of questions for you. There are a whole bunch of them here. The one that I'd begun to ask when you lost your contact was whether specific populations means vulnerable populations in clinical trials. I think it's a great question.
It's a really good question. And actually, all the questions I've seen in the chat are terrific. A special population is really defined because there are special considerations or specific considerations, let me say. Not necessarily because it's vulnerable. And let me make a very important point. Pregnant individuals have been removed as a vulnerable population. Pregnant individuals are adults who can, provide informed consent. And so under the regulations, the HHS regulations, and just recently at FDA, put forward a rule to remove pregnant individuals as a vulnerable population. Nevertheless, the point of the special populations is that there are special considerations that are involved to make sure that these populations can be studied appropriately.
Thanks very much, Lynn. There was another question directed at me, which I can try and take here. The question states, a couple of years ago, leukemia drug was approved via the accelerated approval pathway based on a single pivotal trial conducted entirely in Asia. If a different sponsor wants to take a similar approach, what does it need to do besides making sure the foreign studies are run under strict GCP? So I think there's no restriction on where studies are done. They can be done. We have and will continue to accept data from overseas sites just as we do locally. But obviously the burden for the new drug is the same as for any drug. It's satisfying the regulatory barrier for substantial evidence of safety and efficacy. So although it's certainly possible to do it in another country, and there may be existing data which can be referenced through a 505(b)(2), in the end, the regulatory standard is the same. It's showing that the drug is safe and effective, and good clinical practice as applied to foreign studies is very important. And when a study is performed in another country, it's not unusual for FDA to send out inspectors, just as we do here, to make sure that the standards are good. So I think the points here are really that foreign studies should stand up to the same should conform to the same standards as US studies. And in the end, the bar for an approval of a drug is substantial evidence of safety and efficacy, regardless of where and how it's performed. Let's see. There are lots of questions for Lynn. I have another one for you, which is, are special population studies, e.g. pediatric studies, or a waiver request required for all 505(b)(2) applications? That's a fairly technical question. Any ideas about an answer? Oh, yeah, looks like Lynn, are you still with us? Looks like we're still having problems with her connection. Let me see if I can find one for Karen. Karen, did you spot any questions in the chat that you can fill in with?
Yes, I actually wanted to also address some questions that had been submitted prior to the course. Would that be okay too?
Sure.
Okay. Yeah. So one of the questions that was submitted.
It is, but it brings up, I think, a really important.
Having a lot of trouble with
video. Okay, so one of the questions that was submitted ahead of time was, what is a typical endpoint in drug-eluting stent trials being conducted at the Center for Devices and Radiological Health? And so I looked up one of the more recent approvals, and I saw that a 12-month target lesion failure, defined as cardiac death, target vessel Q-wave or non-Q-wave MI, clinically driven target lesion revascularization, including CABG, was the primary endpoint. And then secondary endpoints could endpoint plus stent thrombosis. And I just wanted to point out that in some of the drug-eluting stent trials, there are usually, or there could be, a subset of pre-specified patients that undergo intravascular ultrasound imaging, both at baseline and at 9 months. And there could be an additional subset of pre-specified patients that undergo optical coherence tomography, both at baseline and at. And I will thank you for that, these questions, and I'll turn it back to Lynn.
Right, let's just see if we're having any more luck with Lynn here.
Yeah, let me try. Can you hear me okay?
We can now, Lynn. We can.
This is really just my apologies again, but let me just say, while I have the minute and my system is still working, that that question about pediatric studies is a technical one, but it's really important because it brings up a more general and important topic, and that is that, again, a tenet that we have I hope to have made clear is that when you think a drug will get used in a pediatric population, then you should plan to study that pediatric population, whether it's a B2, whether it's a combination product, whether it's an NME, whether it's a rare disease. And our laws that are directed at making sure pediatric studies are conducted, the Pediatric Research Equity Act I'm talking about, there's no, for example, well, if it's a B2, PREA doesn't apply. The only situation in which PREA doesn't apply really is when there's an orphan designation or if it's a generic drug. So if you're, and they don't apply to devices. Otherwise, we have the ability to require studies when studies are necessary. And I noticed another question about hypertension and should waivers be, provided for hypertensive drugs? And I'll ask the same question again. Do you expect that this drug will be used in a pediatric population? And if so, we are likely going to ask you to do those studies.
Thanks very much, Lynn. Seeing we've still got you loud and clear, I was thinking you might take one or two other questions. I don't know if you wanted to select them yourself, or I could pinpoint them if you prefer.
Well, I think there were a couple of questions that related to pregnancy and then to geriatrics as well. Mm-hmm. And one of the questions related to geriatrics is, what happens if are geriatric patients approved even if no geriatrics patients were studied? And the answer is yes, geriatric population is an adult population unless there's a contraindication for whatever reason. Having said that, it would be a great failure of our whole plan to improve diversity and to include specific populations were we to approve a drug and have literally no elderly or geriatric patients included. So it's really our responsibility as a whole community here to make sure that these drugs are studied in the patients who are going to be using them, including geriatric patients. And then related to the pregnancy questions, which I thought were also very excellent, the issue of pregnancy is one that everybody's afraid, and not because it's a wrong reason. People are afraid because they don't want to hurt anybody, and we recognize that. And in order to study pregnant patients, we need to have the appropriate non-clinical safety studies conducted first. And if you do that, then I think there is an avenue to consider enrolling pregnant individuals in clinical trials. Remember that the consequence to not getting the data are that the pregnant patients are left to decide for themselves without adequate information. And that's a situation we'd like to avoid.
Great. There were a couple of other questions. A lot of them have streamed in now, but there was one that sort of caught my eye, Karen. The question is how patients feel an objective endpoint such as depression you just mentioned. I think the issue of how a patient feels is obviously something that we're very tuned into, and maybe you want to comment on that as a type of endpoint.
Yeah, so I'm not a psychiatrist, but they do have, and if you have a drug product that works on how a patient feels, functions, that is a valid endpoint. So depression scales are used all the time in depression studies. Thank you very much for that question.
And while we have you on, Karen, there's another one here which says, how does FDA deal with imbalance within a composite outcome? For example, one arm has more deaths while other has more hospitalizations. I think it's a great question.
Yeah, I thought it was a great question too. So thank you. So this is a very, very interesting. So say you had a composite endpoint and you had heart failure hospitalizations and you had death. and the heart failure hospitalizations went in the right direction and the deaths went in the wrong direction. So there were, deaths trended adversely. It would be very unlikely at the agency to get an approval for that, even if you meet that primary endpoint, and that would be a discussion with the particular division. But right, you want to be able to approve a drug product that that doesn't have that adverse harm or excess mortality. Thank you. Leonard, do you have anything else to add on that question?
No, I think that was an excellent answer. We're very close to running out of time. I'm going to squeeze one more in for Lynne, which is there any attempt by FDA to identify special populations based on genotype? A very topical question.
That's, wow. Yeah, wow, talk about forward thinking. At this point, special populations, as I've described in the physician labeling rule, include the special populations I've described. But I think that as far as it relates to special populations that you would be interested in studying, defined by genotype, defined by molecular target, defined by cellular mechanism, all of these are potential areas where I think we are moving towards in the future. So excellent question.
Great. Well, I think we're pretty much out of time at this point. So I just want to conclude the session with a very enthusiastic vote of thanks to Karen and to Lynne, and thanks to the audience for a wonderful set of questions. I'm sorry we couldn't get through all of them, but there may be opportunities later on to answer some of the other outstanding issues. So at this point, I believe we will break and we will regroup in 15 minutes' time at the top of the hour.